Sorry, in this case I was late because I was sent first in the wrong direction with a long way to go all the way around. And I have been in the last time, it was 6 months ago, and everything was empty, so it was easy to orientate, but in this case it was difficult. Thank you all for coming. We have a series of talks, and the speakers, if they may, if they want, they may sit here outside, but it's not so much to be recommended because then you always have to sit like this. So please feel free to sit wherever you want. The only one who has no choice about his sitting is Osman Kaftanoglu, because he's giving his first talk. The floor is yours.
Good afternoon, everybody. I will talk about rearing honeybees in vitro and the effects of feeding regimes on the development of the larvae. Honeybees are social insects, and there are about 30,000 to 50,000 worker bees and one queen bee in a colony, and 0 to several thousand drones. And in every colony during the spring and summertime, if the queen bee lays between 1,500 and 2,000 eggs per day, the larval stage is 6 days, so there is about 9,000 to 12,000 larvae in each hive. Honeybee larvae are fed by the mandibular and hypopharyngeal gland secretions of the nurse bees. And there is no artificial diet for the bees for rearing them in vitro. However, they are fed progressively in the hive by the nurse bees. And during the development of the queen bee, worker bees visit the queen cell, individual queen cells, 1,600 times during the 6 days of larval period. It's about 5.5 days for the queen bees and 6 days for the worker bees. Worker bees are fed about 143 times during the 6-day period. And if we calculate that, they are fed, the queen larvae are fed 266 times a day. That means 11 times per hour. And worker bees are fed about 24 times a day, and it's about every hour they are fed by the nurse bees. That's why it's really difficult to rear honeybee larvae in the laboratory. It's time consuming and it's difficult. There are many efforts to rear honeybee larvae in the laboratory. For over 80 years. The first attempts were made in 1927, 1930s to study honeybee nutrition and test determination. And we are still working on the same problem, even though it has been 80 years since the research has started. There is basic larval diet, which is being used by almost all the researchers. Rambold and Lackner first developed the diet for in vitro rearing, and they used 44% royal jelly, 5.5% glucose, and same amount of fructose, 1% yeast extract, and distilled water 44%. And they were successful rearing these in the laboratory. Then later on, Vanderbank and Chejanovsky used the same ingredients, but changed the formula. And they got good results rearing them in the laboratory. Then Christine Pang at UC Davis, she tested the effects of tyrosine on the development of honeybee larvae, and she used 60% royal jelly, 3% glucose, 3% fructose, 1% yeast extract, and 33% water. But she did not use fresh royal jelly. The previous researchers or teams used fresh royal jelly. However, Dr. Christine Penn used lyophilized powdered royal jelly. So she used 4.2 grams of dried powdered royal jelly, 0.3 grams of glucose, 0.3 grams of fructose, 0.2 grams of yeast extract, and she put 14.6 grams of water, and she made the dye. Later on, our president, Crailsheim, developed the techniques further with his team workers, and they used 50% royal jelly in the dye. And the first 2 days, the larvae are fed with the basic larval diet, which consists of 50% royal jelly, 6% glucose, 6% fructose, 1% yeast extract, and 37% water. But on day 3, they increase the sugar concentrations. This morning, Dr. Robert Page gave a talk, and then he said that the diet composition changes throughout the age of the larvae. So on the day 3, the larvae are fed with a mixture of 7.5% glucose, 7.5% fructose, and also increase the yeast extract to 1.5%. On the last 3 days, they increase the sugar concentrations higher, and also yeast extract to 2%. In the United States, at Arizona State University, we had projects, and we tried to rear honeybee larvae, and we tested the effects of sugars on the development of the larvae. Also, we tested the effects of feeding intervals, and all the other researchers feed the larvae every day. But we developed a technique that we can mass rear them with one time of feeding. We fill the queen cell cups with the given amount of diet and graft the larvae, and we don't touch them. And they develop into pupae and later on adults. In this experiment, we used 53% royal jelly, 6% glucose, 6% fructose, and 1% yeast extract, and the rest of the about 34% water. This was a good consistency to rear bee larvae in the laboratory. And we want to test the effects of feeding regimes. What happens? Can we rear larvae can we rear small bees if we feed them with 100 milligrams of larval dye? Or can we rear really huge, giant bees if we give them 250 or 300 milligrams of dye? Then we had 5 groups of larvae, and the first group received 100 milligrams, second group 150, third group 200 milligrams, fourth group 250, and fifth group 300 milligrams of basic larval dye. And they were in the individual queen cell cups, and all the larvae were fed once during the larval stage, and they were kept at 34 degrees centigrade and 90% relative humidity during the larval stage. Also, we transferred them into another growth chamber when they pupated, and we kept them in the growth chamber at 90 degrees relative humidity. These are the group that received 150 milligrams of basic larval diet, and you can see the larvae, one-day-old larvae, grafted larvae. It's difficult to see, but this is the 300 milligrams of diet. Larvae are here. And this is the incubator or growth chamber. And we have 2 one for larval rearing and the other one is for pupal rearing. After the 6 days period, most of them develop very well. And when you cover it with the cover of the petri dish, they start spinning well. But you don't see them here. And they also seek for food and leave the cell and then go inside the petri dish. We can pupate them in the queen cell cups, but this is too small, and when they become pupa, they have bumps in thorax, and they cannot function well. So we pupated them in the 24-well plates. Okay, this is the survival of the larvae and survival of the adults. 100 milligrams of food is not enough for the larval survival and adult survival. We got the best results with the 200 milligrams of diet. The survival rate was high, and also adult survival was high. 300 milligram was too much, and they drowned in the basic larval diet, or somehow they ate too much and they died. We can only give up to 250 milligrams of diet if we want to rear really big, huge bees. This is the adult weights of the groups. Average adult weight was 78 milligrams in 100 milligram diet, and this is the control group. It was about 115 milligrams adults, because after grafting, I put the comb back into the hive. After 20 days, then I pulled the comb with the sealed brood, put it in the incubator. Next day, I got Neumann bees as the control bees. And their average weight was 115 grams. And when we raised the bees on 150 milligrams of diet, they reached up to 101 milligram, which was little smaller than the natural bees. But when we give them 200 milligrams of diet, they are little bigger than the natural bees. However, when we look at the ovarial numbers, in the control group, they had about 8 or 9, average of 8 or 9 ovarials, but the ovarial numbers increased with the increased amount of diet that they receive. In the 300 milligram group, I had only one bee, which was about 200 milligram, and the ovarial number was around 200. So it was like a queen bee. As conclusions, we can raise bees in vitro with single feeding. This is a big improvement of the techniques. If we feed them every day or every other day, there is a chance of or risk of contamination. But if we feed them only with one feeding, there is less chance of contamination and larval death. So it's a big improvement of the technique. And it reduces the labor and times spent for feeding the larvae in the laboratory. Also, when we are raising bees, hundreds of bees or thousands of bees, it reduces the labor and time. Feeding regime or the quantity of food determines the size of the worker bees. We could get about 80 milligrams of bees or 160 milligrams of bees. These are all worker bees. Control is between them. And the weights and the ovarial numbers are all depends on the nutrition of the larvae or the amount of food they consume. We use this technique on different projects in toxicology, and there's part of the United States that has high selenium content in the soil. So there's honeybee mortality in that area, and we tested the effects of selenium and selenite on the development of the larvae, and for sure, higher concentrations that is found in the soil kills the larvae. We also use this technique in epigenetics and physiology, and we published the papers, and I can give you the publication names. And thank you for your attention, and I would like to make a quick announcement with your permission, please. I will have an Asian Apicultural Association conference next year in 2014 in Antalya, Turkey, and you are all invited, and it will be in a beautiful resort hotel, and I hope to see you all there. Thank you very much.
Thank you. All of those announcements are allowed, but not advertisement for the next APIMONDIA conference.
No.
Yeah, are there any questions?
Here's a question.
Yes.
I noticed that your list of references, they did not include any of the work that's been done by the French Institut National de Research on mass production of bees, like what you're doing there. They've developed standardized techniques where they do this on a regular basis. You might wish to make contact with them to exchange some information, because I think you both could profit by a mutual exchange. And if you wish, I can put you in contact. I'm trying to get them right now to start mass producing drones. I haven't convinced them yet, but it should be possible. And if it is done, which I think it will be done, and that's going to have a very strong impact on future breeding programs and bees, because then you can do all your work during the winter months and everything like that, and then field test either if in the northern hemisphere send to South America, or if you're in South America send to North America. So it should speed up a lot of things in the genetics. So I think a contact with this agency would be important, and also mention that you'd like to see maybe that they start artificially rearing drones. Okay.
Thank you very much for bringing the concept. These are the literature that we published, and we are using a different technique than the European standard technique. We feed them only once, and also this technique is developed in the United States at Arizona State University. But there are other techniques that's being used and standardized in Europe, and it's excellent technique, and I will use it too, because they use 160 milligrams of diet, and they get the perfect size bee. But we have different perspectives. We are studying the development of the bees, and we are not trying to raise perfect worker bee. We like to see what happens, what determines the ovarian numbers. We are really interested in the ovarian development in the worker bees. And the next step will be probably to test the foraging behavior of these in vitro reared bees, or lifespan of these bees. Will they live as long as the in vivo bees or hybrid bees? So there are tons of publications. I need pages and pages to cite all the work that's been done in Europe and or in other countries. There are many research in other countries too, in South America too. And rearing drones is a little tougher, I think, than the rearing worker bees.
I don't know of anything that's been published on artificially rearing drones. You could be the first.
Yes, yes. And I hope there will be some scientists in the audience that will start and we can help. We have other projects and other programs, but it's in my mind, but it's a matter of time. Thank you.
One more short question. Only a short comment. One of the funny stories in science was when we started with this method, there was one recommendation to make a stop of feeding on a certain day, and we tried to figure out what could be the reason. And then the authors told us, well, no, it's just always the Sunday. Okay, thank you very much.
Thank you. Thank you.
The next talk will be from Baohua, and it's about digestion of two types of pollen in the honeybee. Good afternoon. My talk is consumption and the digestion of two types of pollen in honeybee. I'm from China. And third part, introduction. We know different pollen have a very nutritional value for the honeybee. Since the early 1950s, numerous studies have been conducted in the hope of establishing effective scientific method of evaluating pollen's digestibility and the nutritive value for the honeybee. However, the digestion of protein in the pollen of different species is still not well understood. In our study, the digestion of protein in two species of pollen or tested your new factor. The new factor we call the rectum testing method. And the protein consumption and the proteolytic enzyme activity of the milk gut and the pathology of purific memory, short named PM, and the development of the milk gut were investigated in the honeybee read on the two pollen types. In this, the two pollen, this is camellia pollen in China in many plants. This is red pollen. Yes, in microscope. Material and method. We use X method where some bee in this case is still rare in computer. This is pollen guides. We use experiment derived control camellia pollen, red pollen. The control is a man-made bee diet where male and being extracted grease. Use just three kinds of diet. Their honeybee, 3 days, 6 days, 9 days, it's same. Then there are 3 days, 6 days, 9 days, then we stop or hunger the honeybee 72 hours. Then we cut their rectum
we investigated the proteolytic enzyme active of the middle gut and the physiology of PM and the development of the middle gut. Then we use the protein consumption and digestion. The method is rectum cutting method. This is anatomy of honeybee. This is honey stomach. And this part is mid gut. This part is very important to honeybee digestive pollen. Small intestinal. This part is rectum. This part is rectum. When with the honeybee with pollen, 3 days, 6 days, 9 days, then we stop them, hang them 72 hours. Then we cut this part, rectum. The 3 days feeding every kind of diet. 3 days, 6 days, 9 days, then 172 hours collecting the rectum. Then we equalization 3 pollen. Result protein consumption. Total protein consumption by killed bees. The amount of protein consumed by camel pollen diet and red pollen diet were similar at 3 days, 6 days, and 9 days. They are same. The protein digestion. The camel pollen diet group B inhibited significantly higher protein digestion. Here is camel pollen diet. The protein digestion is higher than red pollen diet. And red pollen diet. The middle-gut proteolytic enzyme activity significant difference was observed between the pollen, the camel pollen diet and the red pollen diet. Just the enzyme activity is very different, For the central group B, we read with a mixed mimic diet. Yes. And this is mid-gut PM, the hypertrophic memory. And this part for the honeybee is the absorb capacity is very important. The PM sickness, the bees in the camel pollen digestion group had a significantly thinner PM than the red pollen digestion diet. Here. the PM is center. The midgut thickness, the temporal pollen diet with has a significantly thick midgut wall than the rib pollen diet and the control group bees at each of 3 A's, 3 B A's. This is cross-section of midgut. This is chemical pollen diet. Midgut is PM. This rib pollen diet group B is PM. This is crypt. The crypts of the chemical pollen diet group B's were more closely exposed and had a smaller inside diameter than the crypts of the rock pollen diet group B's and the control group B's. Then we think the pollen and various pollen affect honeybee. Affect honeybee where protein consumption, midgut development, midgut proteolytic enzyme activity. Then role infection affect protein digestion. Our result demonstrated that chemical pollen has a higher nutritive value than rib pollen for honeybee. By influencing midgut development, different bee diets called discrepancies in the digestive and absorptive functions, which is ultimately reflected by differences in protein digestion. Because my English is poor, I don't understand my meaning. Sorry.
Thank you very much. Are there questions in the audience? So let me start with the first one. Do you have an idea what was the difference between the two kinds of pollen? Are there differences in the chemical composition, in the ions, in the salts, in the minerals, in the amino acids, or whatever?
Yes. The control man-made diet contains protein, carbohydrate, mineral, vitamin, and enzyme. Yes. I think that this is the meaning this study is tell beekeeper if we have $1,000 US, which pollen is better for beekeeping the honeybee. Yes.
So for some pollen, we know that there are some amino acids in a too low amount, so they are lacking a certain amino acid. Is this true for one of your pollen?
Yes. If we lacking some pollens, we can choose. I think high evaluated pollen, for example, the chemical pollen.
Thank you. Any more questions?
Yep, one here, on the wrong side of the table. How do you think this thickened midgut will have an effect on resistance to certain bee diseases? Do you think that in that case, if you have a reinforced membrane there, that you'll have less susceptibility to certain diseases?
In this, the honeybee is reared in the cage, incubate.
So some parasites are penetrating the gut wall. Do you think that the thickness of the gut wall
Yeah.
is inhibiting some pathogens to enter the bee?
If the thickness is the PM is sick, is sick, is good.
That's one word.
Dear Mr. Ying, is it possible that I receive PowerPoint and USB?
The question was if you would give him the presentation on the sticking afterward. I think you should communicate in the break. Okay? Yeah. So try to contact this guy, please. Okay, thank you very much. Thanks. So may I announce the next speaker, Greco, the only one who wanted to sit beside me. The development of an infection model to study how Trophallaxis insects on pathogen transmission in the Honeybee Apis mellifera.
Yes, thank you. My research is based at the University of Bath in the UK, but there we work in conjunction with the National Bee Unit at Vitro. And at the moment we're working on the epidemiology of European fowl brood. So during those experiments, we thought we should look at the way pathogens are transmitted from one bee to another bee, from one beehive to another beehive, and from one apiary to another apiary. To do that, I tried some interesting techniques, and one of those techniques was to actually put a beehive in a computer tomography X-ray scanner to look at bees and to see if we could trace any sort of food exchange or pathogen transmission via Trophallaxis. Thinking that Trophallaxis might be one of these mechanisms where you can pass pathogen from bee to bee, beehive to beehive, and apiary to apiary. During those experiments, interesting patterns emerged when we labeled food. This is a cross-section a scan through there, through the hive. You can see the structure of the comb, and you can see definite patterns occurring. Blue area here, green areas there, another blue area there. So blue honey was honey that wasn't labeled or marked with any material, and the green honey was honey that had labeling material, essentially iodine. So we saw these patterns happening. But interestingly enough, that when we provided food inside the hives, that's an Apidea hive, inside the hives, we had labelled food on one side of the feeder with a partition down the middle. You probably recognise these feeders. Again, the blue honey or syrup, sugar syrup was unlabelled, the green was labelled, and the bees could feed on that ad lib. There was no restriction, they could choose to feed on the marked or unmarked syrup. And what we found, or what we observed, was that some of the bees were collecting the labelled syrup, and some of the bees were unlabelled. Even after 7 days, some of the bees did not have any labelling of them. This was quite interesting. So we thought we should look at this a little bit further, and we collected bees at random, hive bees at random, and we put them in little plastic segregation cups, little plastic cups, and we fed them with a little feeder at the top with labelled food, and we put 20 bees in at a time, and we ran the box through a scanner, and even when we have only 20 bees in each cup, occasionally, even when you only had 20 bees, some of the bees were not taking up the labelled food. We found that even with 20 bees set up, we had actual bees collecting the food and feeding the other bees. And some bees, they would set up this sort of hierarchy. Some bees would automatically become receiver bees, and some would become donor bees. And the donor bees, even though there's only 20, were ignoring some of the receiver bees, were not feeding them, even though they were begging. And those bees were dying. They were unmarked and dying in the cups. This was interesting. So we set up an experiment to see whether we could actually trace this, to establish whether there would be any pathogen transmitted by Trophallaxis. So we had to set up an infection model, and we used pathogenic fungus, Bavaria bassiana. Normally, as you know, Bavaria is throughout the colony. It's not so damaging to bees, not so pathogenic. But if you give the bassiana in high amounts, it becomes pathogenic. And these amounts are not biologically usual. We have to increase the amounts before it becomes pathogenic. The reason for that is if it was highly pathogenic, all the bees would die and the experiment would finish. Second thing we wanted to do is to see whether we could test whether bees actually preferentially fed within a hive. Selected some bees over others. So this is the method we used, and this is the low technology plastic cup, about 250 mils in size. It's a plastic cup, nothing special. But this, again, another piece of low technology. This is the bit that's unique about an infection model. Essentially, again, it's a plastic partition here and a perforated section at the top. And it has a perforated section at the top, non-perforated section at the bottom. Why is this significant? You can put either feeders here or breathers, depending on what your experiment is. And once you do that, you can put red bees in the top, blue bees in the bottom on that section, and green bees on that side. Now, which means these can be separate groups, all within the one segregation cup. And of course, these groups could be any groups you decide to use in your experiment. These could be foragers, these could be receivers from a different colony, these could be receivers from a third colony. It's up to the scientist as to what you want to put in there. But this T-piece is the thing that enables you to do that, which hasn't been done before. And we have a black screen. I have no idea why we have a black screen. That's back again. Oh, there you go. Sorry, that was me. Okay, so this just shows up that shows that these bees can come in contact trophallactically with these bees, and these bees can pass food backwards and forwards this way, but these bees cannot. Okay? So our experiment was this. We put bees from hive 1, H1, in the top, and these were all forager bees. So we collected bees at the entrance coming back with pollen on their legs, definitely foragers. These were hive bees selected at random. We don't know the ages of these bees, they were on the comb when we collected them. And before I move on, the way we collected them was with the vacuum cleaner. So that we could put a tube in these holes, and it creates the vacuum, and you can just collect the bees. You only have to handle them once. In fact, you don't actually handle them. They go straight into the segregation cups using a vacuum cleaner. And we had receiver bees from a second hive 2 in that section. Then we swapped that around with hive 2 bees, hive 2 foragers at the top, hive 2 receivers, and hive 1 receivers on that side. And we introduced 60% sucrose, ambrosia, so the 70% with Bassiana, Bavaria Bassiana pathogen. So that meant that these bees were the only ones that had access to any food, actually. They could just feed themselves at it. They then had a choice of whether feeding or not feeding, both or neither, or just one. And we just did many, many repeats. So once you have them in the plastic cups, you just do many repeats to increase the robustness of the statistics. And the results. What we measured was survival of receivers. And the experiment ran over 10 days. Most bees died after 7 days. So essentially, the experiment finished after 7 days. But at day 4, we could see that hive 1 survival in the receiver here had very little difference, 3% difference. And hive 2, when we had hive 2 foragers, we had 7%, which wasn't all that significant, it wasn't significant, but then at day 6, the trend continues with only 1% different survival between this group and this group. When we had hive 2 as the forager feeders, we had a larger difference. Hive 2 receivers were dying much quicker. And at day 7, that trend continued. Again with hive 1, there was 8% difference in survival rates. It was not significant, and highly significant when we had hive 2 foragers, feeders, with a 21% difference. And just to show it graphically, these are the results that displayed graphically. Not much happened before day 4. Bees started dying, dropping off at day 4. We can see the 2 control groups behave fairly similar. In both the group that had the hive 1 forager feeders, quite similar to the hive 2 forager feeders, the controls are quite similar. And we can see with the hive 1 feeders, we had an 8% difference at day 7, which wasn't significant. But with the hive 2 forager feeders, we had 21%, and it was highly significant. So we didn't measure spores, Bassiana spores, but this was an indication that hive 2 receivers, or the bees receiving food from hive 2 foragers from their own colonies were dying much more than the bees from hive 1. So the results from that experiment are significant for pathogen transmission because we only tested 2 hives, but there was it looked like there was a preference where hive 2 foragers were preferring their own receivers, their own bees. They were ignoring hive 1. Bad news for the receivers because they were getting lots of pathogen in this experiment. Whereas hive 1 didn't show any preference. Why is that significant? For heterogeneity within a colony. Most of us should know that irrespective of patronage, honeybee colonies can contain over 40% of bees from other colonies, worker bees from other colonies. And if you have a colony with that has a preference for their own then they'll be feeding their own more often than feeding another colony, and that has an implication if what they're feeding them contains pathogens. So the other thing that is significant is that preferential feeding can be managed by queen breeders if so desired. This preference, this phenotype, this preferential feeding stems from the genotype of the queen and the drones that mated with that queen. So there's an opportunity, if we continue with these experiments, there's an opportunity for breeders to actually select towards these traits or away from these traits if they're desirable. So future research. What about the drones? I'll try to go through this. I think we've got plenty of time. Okay. The colleagues in York have discovered that drones contain large amounts of EFB spores, which is quite surprising. But they actually, you grind drones up and you can run them through lateral flow devices, and there are huge amounts of EFB spores within drones. So the drones are always mostly forgotten in lots of experiments. However, what we did is we placed drones and worker bees in experimental cups, segregation cups. Worker bees from hive 1, worker bees from hive 2, drones from hive 1. So that just follow the colour patterns. And what we did is we saw that worker bees were feeding their own drones. Not surprising, that's what worker bees normally do. But then we physically drifted drones from hive 1, put them into the top segregation section, provided some food, and surprisingly, drones were feeding workers. So the other way around. They were feeding workers from another colony. They were also feeding these workers. But the important thing is that they were feeding bees. If we imagine that these 3 cups are 3 separate beehives, then we have a transmission route. The workers from this colony occasionally visit other colonies. Sometimes we have 40% in other colonies. And they feed drones. And they'll feed, most workers, foragers will normally feed anyone that comes towards them. And interestingly, the drones actually feed their own workers. We have videos and footage of this if anyone's interested. We can actually see it happening. What we can see is drones opening their mandibles and workers palpating with their tongue. So the biggest surprise was that drones, if you put 3 groups of drones in these cups, they start feeding each other like crazy. Not like crazy, but about 20% of them will feed each other. So drones will, in these experiments, drones are actually feeding other drones. So it's easy to watch this happening, but what we tried to do is we actually marked the food and we ran some more VR scans, some more computer tomography, and we then euthanized the bees. 3 minutes, okay. And we actually put the white bits in this image are actually the marking material, and you can actually see the food in these groups. So these are drones feeding workers. That marking material is actually food that's gone from drone to worker, and the same with these other groups. See, every second line should not have marking material in them, but it does because the drones have been feeding. Thank you.
Thank you very much. Some of the results are really surprising. Questions? Yes?
What was the radio label? What compound?
The question is, what was the radio labeling? We're using X-rays for this technique, so we don't actually have to use any radio labeling. There's no radiation given off. It's iodine, and it's just a really heavy element, and that makes the element show up white on an X-ray.
So why did you decide to use the pathogen you used?
Yes, we used the Bavaria bassiana because it doesn't kill the bees unless you use large quantities. And that way the experiment can continue and trophallaxis can continue without all bees dying. So it's mildly pathogenic.
And were all the bees fed in all the 3 compartments or only the ones on the top?
So only the bees at the top have access to food. Any bees at the bottom needed to get that food from those forages.
So couldn't some of the bees of the foreign colonies have died because they had not been fed?
Yes. I was hoping you wouldn't ask that question.
Other questions? Yes?
How is it possible that the drones feed worker bees? That drones feed worker bees? How is that possible?
How is it possible that drones feed worker bees?
Yes.
They open their mandibles, regurgitate.
Because you showed that drones feed worker bees.
Yes.
But
How is that possible?
normally the bees feed the drones.
Correct, yes. The question is that normally the worker bees feed drones. And this is a very good point. This is what this experiment is showing. If you remove worker bees, if you remove access to food, they get hungry, they start begging. And through the mesh, they're sticking their tongues up all the time. And the drones come along and they open their mandibles and the bees get a little bit of food. And the better answer to your question was, that's why we did the scans. If you see the contrast in the bee, you know that it's died from pathogen, not from starvation.
And the larvae and the drones.
Yes.
Do they feed the larvae?
We didn't test whether drones feed larvae. That's a nice question. We didn't test that.
Well, I would think no, because the larvae don't just need sugar water or things like this, they need protein, and the drones are not able to distribute protein. But to tell another interesting fact to this, I've already also seen queens feeding other hungry bees, so it's also the other way around. Any more questions? Then, Mark, thank you very much.
Sorry, it's my blind eye.
I think the guy behind you was asking first, sorry.
Well, I understood that if you separated drones out in a cage because you wanted to use them for artificial insemination, that they actually become sick after an hour or two because they haven't got contact with workers.
Yes.
But if you were feeding them, then they could look after each other, or do they get some other benefit from making this contact with the workers?
Yes, especially the statement was that drones, after an hour or so, by themselves, get sick and die. I have to say that they are quite delicate. So on occasions you could collect 20 drones for the top section, and within half an hour they're all dead. You don't know why they're all dead before you even start the experiment. But if you do want to run experiments with drones, it's better to, and you need to keep them for some time, it's better to include workers with food, because the workers are the more active ones and they'll keep feeding the drones to keep them going.
Besides other parameters, you measured the water intake of the workers, but the aging of the workers wasn't the same. Don't you think that
Can you speak louder, please?
Yes, I
hear
did.
you.
He asked whether we knew what the age of the workers were.
for the future experiments?
For future experiments.
Not better to use
Yes.
the workers of the same stage.
If you do lots of repeats, lots of numbers, then you can collect workers at random, which evens out any differences. But you need to do lots of repeats. So the other thing, the way to do it is what you're suggesting is to actually know the age of the workers when you collect, and they might act differently. Yes.
You treated this by the quantity of the repetition.
Yes, lots of repeats. Yes.
Thank you very much.
So I have a series of announcements at the moment. One is, please don't stand here behind. There are several seats on this side available. First, the next talk that please say is working by Peter Neumann, and he's not coming from Brazil. As is indicated in the program, he is coming from Germany. The next point is also related to him. So there will be a e-book presentation in the break of this meeting, so it will shorten the break. But I think the break is anyway too short to do something else. Also another surprise regarding this. And I think it's the third or the fourth announcement now. In the later afternoon after the coffee break that might not exist, Neumann is so kind to take over the chair because I have to go to an APIMONDIA meeting. Last announcement, the talk after Peter Neumann will not be given by Goran Mirjanic, but by Greco Tomljanovic. Okay, so now it's yours.
Good afternoon, ladies and gentlemen. Everybody can hear me? Is that all right? Please, one of my mistakes is I talk very fast. So if you have problems with the translation, because I talk too fast, raise your arms and I try to speed down. Louder, all right. Okay, so then I have to sit like this for a few minutes. Okay, all right. First of all, I would like to thank very much Crailsheim for organizing this symposium and for giving me the opportunity to speak here today. As you may have noticed, there are a lot of names on the first slide, and in fact there could be many more, so that you can't read it properly. So here what I'm going to talk about is a work of many people working together. So a picture kind of provided by Anna Gaida. It was not working, but see it in the right corner. Basically showing our last meeting here in Ukraine, and we're going to present a big group effort, and many people contributed to what I'm going to present today. Let's talk about honeybee colony losses. Surely it's a problem. It's not nice to have that colony, so there are a number of issues to be addressed. And number one is we need field data on colonies lost in the field. So we have to know exact numbers. What is the proportion of colonies dying in certain areas? That's clear. We need raw data on that. Second, we need research on causes and mitigation methods. We need to understand why colonies are dying and what we can do about it. And then an important part is dissemination of results to the stakeholders, meaning if we have a breakthrough and discover some new method, you should, as beekeepers, should know about that so that you can actually use it. But there are also stakeholders, important ones. For example, decision makers, policy makers, grant awarding agencies, there are a whole range of people involved in bee health, and they should know about that. Good. Here we're having a picture showing a map of Europe with loss data over 2 winners. And as you can see, oh no, it's working now. And as you can see here for 2 winners, as you can see here, losses in Turkey were about double the losses in Greece. Serious. Well, no, you can't say that. It's not possible to say that the losses in Turkey were higher than the losses in Greece. Why not? Because here you compare apples with bananas, or whatever, muffins with donuts. It's not possible because the data have been collected in a very different way. And to give you a further explanation here, if you look at country X, I don't want to tell names here, in country X, 2 beekeeping organizations provided data on losses in one country, or big ones covering the whole country. And the losses were 52% in one association and the other 6%. That shows you can't compare the numbers and there's a need for standardization. You have internationally standardized data that you can compare Greece and Turkey actually, and also from the beekeepers, and are required really to compare losses between countries and years. You need standards here. And here we go. One of the other major efforts of the Carl network is to get such standardized data. Again, a map of Europe. Here you have winter mortality, and for the darker the color, the more severe the losses. And here you can compare. Here, for example, losses were more severe in the Netherlands compared, for example, to Italy. Having different winners, different years, different colors here. Very severe losses in Macedonia this winter. And it shows different maps that we have clear and standardized evidence for high losses. What are high? I would say roughly above 10%. That is, 10% is kind of normal, but above 10% is high losses. and over several years. So it's not happening in 1 year and the other years are fine, it's happening in several years over time. So your input from the beekeepers is crucial for that, to show you that data. We are only able to show the data because many of you have helped in the past and are still helping to collect such data. So that is a questionnaire by Roman Fanetze and co-workers developed in Carl's working group 1. We're having a number of simple questions, and Charles says here we answer certain issues. So your input is very important, otherwise we couldn't show you that. And the losses are variable in time and space. You know, sometimes you have a country having high losses one winter, next winter low losses, and they're variable. And that clearly shows there's an obvious demand to understand drivers of honeybee mortality, so why colonies die. Good. So life is complicated. Cut a long story short. Well, all of you know that Varroa is certainly a major problem, but if you talk with beekeepers doing everything correct, Varroa control the formic acid, oxalic acid, according to protocols, still colonies may die. That is really bad. So there are more factors than Varroa can kill colonies. And here all of those, nutrition, pesticides, well, we don't have to talk about that, it's clear they're not healthy, and genetic diversity and beekeeping, all of those factors by themselves can actually lead to colony collapse. And to make it more complicated, factors may interact. Good example, Varroa mites and viruses. Varroa may act as a very potent vector of viruses, and that together can be a killing factor. And to make it even more complicated, or the between groups of stresses interactions are possible. For example, there are a growing evidence that pesticides, colonies poisoned by pesticides, may have more of a problem with parasitic fungi and Leucemia disease. And it's getting very complicated. And so what's going on here? For an individual bee researcher, for one bee lab, it's really, it's very hard to do that. There are so many factors, so many issues to be addressed, concentrations of pesticides, different feeding regimes, and so on, and so it's very difficult. Maybe we have to ask for some help. So maybe Super Bee Man can help us out here. Well, most unfortunately, as you understand, he is very busy in saving the world, so you may not have so many time for saving our bees. So maybe we can go without asking Super Bee Man to help us. So maybe join forces, and indeed, we are certainly not super bees, but we're learning from the bees and working together, having the colors, a hive basically, like a colony, with a division of labor, learning really from the bees, division of labor. Some specialize in monitoring and diagnosis, others specialize in pathogens, or in the beekeeping or genetic part. So by joining forces, working together for a common goal, we can achieve that. And it's a great team here over the past weeks, years, several years actually. Skills committee and people are arranging that network. So, and what are achievements over the network? Of course, you can meet in great places like in Athens, having a beer together, enjoy, but what is output? What came out of it? And so network and capacity building is really our bread and butter. So we organize meetings to endorse, to facilitate research. We had conferences, workshops, short-term scientific missions and training schools. This is actually a great tool. You can send students to another laboratory to learn a technique for a couple of months, and that has been proven, I think, very useful for doing research. Next part is coordinated large-scale experiments. And here's the underlying reason is honeybees are special patients. I'm a veterinarian now, and if people dealing with cows and chicken, they have a single animal. Single animal is sick or not sick. A colony, honeybee colony, is very different. You're having a single bee that's an individual insect. It can be sick or healthy. But at the end of the day, you have the hive as a unit. The colony is our patient, it's a functional unit. Some workers may die, but the colony still lives. On the other hand, colonies may buffering very well, and suddenly they crash. So here we have to understand the colony level. That makes it very difficult. And nobody can run experiments with like 1,000 colonies or 600 colonies. It's very difficult. So joining forces and running experiments together, I believe, is a key if you want to understand bee health better. So an example, I think a very good example from Colors is a genotype environment interaction experiment where that happened, where different laboratories used the same method to do in one project. And we had an N from 621 colonies, which is amazing. No laboratory can do that. 600 hives, that's really great. So the colonies were transferred across Europe, here from Helsinki to Bologna and vice versa, and tested for their ability to survive. Great one, a couple of papers coming out of that, if you're interested in. There's a special issue of JR, and we'll have open access, so you can just download it from the net. And that, I believe, is a central mechanism for us as scientists if we want to understand better bee health. Joining forces and running large experiments, let's say with 10 or 20 groups across the world. Good, it's not easy, but it's doable, as we have a proof of principle. And then updated standards for research and monitoring. I told you already for the monitoring, if you want to compare data between the United States, South Africa, and China, you need to have a common protocols, otherwise you can't do that. And the same is true for research. If you're having different results on the famous pathogen X in one European country, and in other countries you have other results, there are a lot of reasons why that is the case. It could be different pathogen virulence, it could be different ability of the host to survive a disease, but it could also be that different methods have been used. So to avoid that, standardization of the research is also an important point. So here we have a Carl's bee book, an editorial, and well, what is a bee book? The bee book is a manual. Here we have 2 volumes out there. Vincent, Dietemann, Geoffrey Williams, Jamie Ellis, and myself editing it, and where we address such issues. And it's actually big. We have 37 chapters, more than 200 authors, and 1,700 protocols. And these are practical guidelines. So you take the book, you read it, you can take it on the lab. You need so-and-so bees, so-and-so whatever sucrose, and you do the experiment step by step. So I would have loved to have such a manual when I was a student, but well, now here it is. And also, it's also relevant for beekeepers. If you're interested, for example, in queen rearing, you find a very detailed protocol, easy to understand how to do queen rearing. So I think it's amazing. It's really nice to have. And so I can recommend you can either buy the hard copies, or all the articles are open access. Go to the IBRA website or go to the Carl's network and just download the articles. For free, you can use it for whatever purpose you want. Good, then fourth, dissemination of results to various stakeholders for bee health. I told you at the beginning that if you have results, it's very important. Not only to tell our fellow scientists in papers, hey, you found something interesting, but also to the people like you, the beekeepers, or to veterinarian authorities or the politicians, that something has happened and that they're supposed to know that. So we're having a whole range of different tools from the website and flyers to actually inform people. Good. Cost action is over now. It was almost a year ago. And money makes the world go round, also from colors. So we were like, what's difficult, how to find sources, and it was a bit of a transition period, but we have definitely decided we carry on. We create, just over here in Ukraine, we managed to do that. We had a Colors 2.0, a complete reboot of the system. And we have now a global association to promote bee health. We have a legal status, like a charity organization or a non-profit organization with a bank account and a postal physical address in Switzerland. We're having here a team of Karl and myself. We have stayed in the president and vice president, but we have also like some members from the former executive committee, but I'm very happy that a lot of fresh input came into this committee. So here, for example, Pathogen from Thailand is new in there, and Victoria Soroka from Israel. We also have not only changed the way how to the legal status and how we govern it, we also changed how we approach it. We are not anymore having these strict boxes on doing things. We have a new flexible system, and we have so-called task forces. And I just want to give you, it's work in progress still. We have to decide where we put more emphasis on. But just an example is the varroa control. That is very relevant for all of you in the field, how to treat varroa correctly and properly. Is an easy manual to use if you want to use formic acid and oxalic acid. There may be differences between Norway and Italy and Turkey and Israel. How you can do it and why and not, what a side effect. So here I think is a great need for improved varroa control. There will be a task force as an example. And also like projects I told you about the large coordinated projects. And one is here, CSI Pollen, crime scene investigations. Of course, you may know CSI, whatever, maybe CIS and some other issues. And CSI Pollen, so we have crime cases here, maybe we have. Indeed, here's CSI Pollen. I introduce you. It's a project from Joseph van der Steen and Robert Roach, and I initiated. And it's taking advantage of beekeeping and helping you in collecting data. So the citizen scientist investigation, that is the CSI, the acronym, the citizen scientists are you, the beekeepers. You're collecting pollen in the field, using standardized approach, and collecting it, and you're getting the data. And that is, I think, is nice, because here we can look at the pollen diversity of forage that is available to honeybees here based on the different coloration. And pollen is so important, we have talked already here today. So, and we're sure that this, again, here is a principle. What I told you about the different, well, coordinated experiments in different laboratories across Europe, that citizen science is something we have not really used or to a great extent. And I would be delighted to see that happen. That from the beekeepers you can data, and we can use that to help you in telling you, wow, here's an important point for bee health. Now I would like to give you a summary. First of all, we have clear evidence for high losses over several years in many countries in the Northern Hemisphere. That is clear, and it's time to do something and change that, promoting the bee health. International comparable loss data are important because, again, if you want to compare data from China with South Africa and New Zealand, you must get the data in a common way. Then there are many factors for bee health and interactions. In fact, there's an infinite number almost, I would say, of interactions between pesticides and pathogens and whatever else is out there. Good, so networking is urgently required to get standards and working together. And the bee book is here an example as a manual, not only for scientists, but also for interested beekeepers. So from you, if you want to do, for example, breeding, you can use a protocol, the COLOS protocol on breeding baits. Then the COLOS 2.0, just told you, we had just a restart a couple of days ago, and we have a new association to promote bee health, like a charity organization. And we will disseminate the information about our startup properly. There will be a press release by IBI actually later to that. And we have now new task forces. We don't think anymore in the pest and pathogen boxes or the pesticide boxes. We open it up and we'll be addressing issues of interest. And for grow control and breeding, with, for example, 2 task forces, we will start right after this meeting. And also new large-scale projects. And the CSI Pollen is one advantage, one example on how, for example, US beekeepers can help us to better understand the scientists' bee health, and this will also help you. So it's a win-win situation for both sides. And I just want to highlight these 2 issues here. Here in this part, international comparable loss data, and those citizen science projects, we would ask you for your kind help to help us in doing that. So collaborations with the beekeepers are an important cornerstone for the COLOS network and for our efforts to improve bee health. Good, so the new initiative, I think it's a very promising avenue for bee health. I'm actually convinced. And I'm very excited about that. It's really nice. But I would like to thank the Ricola Foundation for supporting us, cost, and we had also a meeting financed by the Bee Dog. And then, of course, I would like to thank my fellow network members. It was very fun and productive to do that work. And last but not least, I would like to thank you for your kind attention. Thank you.
The talk is open for discussion. Yes.
Is Colossus able to achieve funds as a unit, or is it still up to individual research establishments to collect their own? Because if you had funding yourself, then you could have some control over research, possibly.
Colossus is an independent charity organization, or like a non-profit organization like Greenpeace.
Yeah.
But we have a kind of a limited scope. We focus on honeybees and honeybee health. And to answer the question, yes, we can receive funding, and we hope to receive funding from very different sources. So in the audience, if you are aware, of the way to support us, that would be great. And we don't talk about, it can be little money. For example, the crowdsourcing principle, I guess. So you ask around, like it's a very nice way for bands not to publish their albums. They ask everybody, please, the fans, give some money, 2 or 3 years, and you help us. Yeah, so please, if you're aware of money, I will announce it. And it will not be under my control. We have an executive committee, and the board decides what to do with the money. It will not be, although the president, I have not the saying, I cannot decide myself certainly how to spend it. It's a group decision.
If you want to will some money, we are all ready to give you some.
Thank you very much. I'm very happy to hear that. Great. Thank you very much indeed. I'm delighted about that.
Other questions? No? So I think we will...
Oh, yes, please.
Kolos goes only about honeybees, but the type of honeybees most of them are solitary bees. Solitary bees.
Yes.
They have no flora. They have no beekeepers who don't treat for their hives not well. But also there are lots of collaboration with beekeepers is of course good, but is a collaboration with the solitary bees.
All right. Okay, thank you. The issue was raised, which I think is very important by the gentleman over there. What about the wild bees, solitary bees, but also probably bumblebees? So what's going on with them? Because they don't have beekeepers to take care of them. Yes, indeed, that's a very, very important point. We have decided in Kolos that we focus primarily on honeybees, not excluding wild bees, but we focus on Western honeybee. But there is an initiative, an ongoing initiative as a cost action, very similar to like actually the identical mechanism how Kolos once started in 2009. There is now a proposal that's called Super Bee, where the pollination itself is the ultimate goal to ensure sustainable pollination across Europe. And there, the wild bees play an important role. So within the super bee cost action, the honeybee people and wild bee people will interact. And I think that's a great opportunity because you have a bit of complementary expertise. In the honeybee field, we tend to have some deeper understanding of, for example, pathology, while the wild community knows about landscape ecology, for example, probably better than the average honeybee researcher. So yes, there is an ongoing initiative, and that should be taken into account. I absolutely agree with you.
Thank you very much. You will see Peter twice again today, here on the floor. So the next talk, as I have announced, it's a change. Goran Mirjanic is not here, and Zlatko Tomljanovic is giving his talk.
Actually, first of all, I would like to say hello to everyone, and also thanks to you, Karl, to give me the opportunity to jump in as a replacement and give a talk instead of those authors here. All of them are good friends of mine, but they couldn't come to Kiev. They are very busy working on some project in Southeastern Europe, and I will give a talk instead of them. Well, when we talk about health status of intestine of honeybee, we have to tell that honeybee meets their needs from the four natural resources: nectar, honey, pollen, and water. Sometimes there is need of supplemental feeding of bees, which is prepared in two ways, using acid and enzymatic hydrolysis of sugar. Both of these chemical processes are in the service of small physiological absorption of honeybees, given that it uses only simple sugars for its own need. And also, residual acid in this food causes splitting of its intentional tract, with significantly shortening of bees' life. And also, as we already know, that enzymatic hydrolysis of sugar with the help of enzymes, sugar inverters, is a slower process, but without and harmful effects of life of bees. Let me talk about something about material methodology. The study was carried out on the experimental apiary in the northwest part of Bosnia and Herzegovina. Experimental cages were filled with 200 bees, which were fed with 2 netted holes with prepared foods according to experimental design. In the experiment, we used the following sources of bee food as a control group. In a control group, we used acacia honey, and also we used sugar syrup, acidic invert syrup, used tartaric acid, and enzymatic invert syrup. And experimental groups of bees were fed in following variants. That only with syrup, that only with syrup plus 40 grams of brewers used. That syrups with 40 grams beer wort, and that syrups with 20 grams brewer's yeast and 20 grams beer wort. What were the objectives of the work? Were to determine the optimal feed source for bees as honey substitution, the effect of different feed to midgut epithelial layer of bees, and the effect of different types of feed to length of their lives. Let me go quickly through the results. On this side, you can see the middle gut of bee fed with acacia honey. It's a normal honey. It's a normal food for bees. And on the picture 2, you can see the middle gut of bee fed with sugar syrup. So the results with on a control group who were fed with acacia honey, that the epithelial cells of the midgut are quite conservative, high and are closely adherent to each other. But on that other group, experimental group, the midgut wall is properly wrinkled and in close contact with intestinal content. And also, epithelial cells are properly arranged on the inside intestinal wall. When we talk about intestinal contents, it's quite homogeneous and compact and fit close to the intestinal epithelium, which suggests well-digested food, like in natural. But we can see here that in a group fed with sugar syrup, slightly damaged top layer of epithelial cells, and which is not present in all cells, and intestinal contents is not homogeneous, fairly loose consistency, and lies directly on the intestinal epithelium. You will do this. The next one, the next experiment was carried out about middle gut, what was happened in the middle gut of the bee fed with sugar syrup and yeast, and also with middle gut of the bees fed with sugar syrup and real wort. As you can see here, occurrence of spacing between individual epithelial cells and their not homogeneous distribution. On the other side, you can see here quite stretched bone wall leading to its reduced absorptive ability. And also, as with the sugar bath, sugar that intestinal content is not homogeneous, fairly loose consistency and directly next to intestinal epithelium. And on the other side, epithelium cells have damaged surface, while one part of the high epithelial cells missing, with only regeneratory cells visible. And also, when we talk about intestinal content, in a group fed with sugar syrup and beer wort, you can see down on the right side that the content is not homogeneous, very loose consistency, and lies directly on the intestinal epithelium. Also, on picture 5, you can see histological view on a middle gut of beef fed with sugar syrup, yeast, and beer wort. And on the picture 6, middle gut of beef fed with acid invert syrup. So, on that group fed with combination of sugar syrup, yeast, and beer wort, they found bone quite stretched and thin, so transverse wrinkles are not visible. On the other side, when they give this acid invert syrup, epithelial cells are widely separated, and there are a number of gaps between them, which are manifested in the form of deep indentations in the epithelium, and also pronounced big damage of epithelial cells with their lack of intestinal epithelium due to the damage. But in a group with acid invert syrup, almost totally damaged high columnar epithelial cells with only laboratory cells not checked. And when we also finally talk about intestinal contents, lies on the epithelium with a good portion of the content compressed in the center of the intestine, but in a group fed with acid invert syrup, the content in the intestinal lumen is a rather loose consistency with visible parts of disbanded cells. So help me. And on picture 7, you can see also histological view on middle gut of beef fed with acid invert syrup plus yeast. And on picture 6, middle gut of beef fed with acid invert syrup and beer wort. Results a similar situation as with acid syrup, but with epithelial cells better preserved. In group with fed with invert syrup and beer wort, complete layer of epithelial cells rather damaged by death, but fairly compact and in close contact with intestinal content. And also, in group with invert syrup and yeast, there are sites of observed damage, which cover almost the entire depth of the epithelium, but on the right side you can see intestinal column content of rather homogeneous consistency. And finally, the results on picture 9, middle gut of beef fed with acid invert syrup, yeast and beer wort. And on picture 10, middle gut of beef fed with enzyme invert syrup. So about almost 2/3 of the total thickness of the epithelial layer is damaged on the entire surface of the lumen. But when they use and administrate to this enzyme invert syrup, high columnar epithelial cells properly aligned on the basal membrane of the transverse corrugated pipes midgut. And also, when we compare some damage of epithelium on this group damaged epithelium is a quite compact and homogeneous composition, but on the other side you can see partially separated peritrophic membrane, which wraps some slightly loose bowel contents. And also we got on picture 11 middle gut of beef fed with enzyme inverse syrup and yeast, and on the picture 12 middle gut of beef fed with enzyme inverse syrup and beer wort. Results on the left side, you can see that the slightly damaged surface epithelial cells and thereby expressed regeneratory cells, but on the group fed with enzyme inverse syrup and beer wort, in some places the epithelial layer is totally damaged and to the muscle layer, and also the intestinal column content in the lumen is free of obstruction and directly, as you can see here, adjacent to the midgut epithelium. In that other group, the residual content in the intestinal lumen organized in a homogeneous cluster. And also on picture 13, you can see middle gut of beef fed with enzyme inverse syrup, yeast, and beer wort. Epithelial cells are deeply damaged through all the gut's surface, and when we talk about intestinal contents, is compressed into a homogeneous mass within the gut lumen with a belted one homogeneous layer, and it is difficult to digest for this. Also, you can see here a table of lifespan of bees. That's a feed type here from honey, sugar syrup plus yeast, all the way down to the enzyme syrup plus yeast plus beer wort. The experiment was conducted for 3 years, and the average lifespan of bees, of course, was the best 27 days when in the control groups. But the second one is this one, the group fed with enzyme syrup. Of course, group fed with acid invert syrup, the lifespan, average lifespan was just 12 days, and it means that it suggests that it's not very clever to use acid invert syrup in honeybee feeding. And let me give you a conclusion. Based on our research, it can be concluded that feeding with different food sources has different influence on the digestive tract of bees, especially in the midgut epithelial layer. And natural source of food for bees, honey had no harmful effects on the midgut. As you can see, the average lifespan was 27 days. Epithelial layer and the intestinal contents were completely attached to this layer, which leads to the quality of digestion and maximum nutritional absorption. But similar results were got when feeding bees with sugar syrup and enzyme inverted syrup without the addition of yeast and mold. This means that each addition of yeast and malt led to the damage to the midgut epithelial layer, and the difference arises depending on the, of course, food source. The most serious damage on the epithelial layer was found in the midgut of bees, as we talked on the previous slide, fed with acidic invert syrup. And regarding the impact of the different fed on the length of the live bees, we can conclude that it can be concluded that feeding with honey enzyme invert sugar syrup had a positive effect on the lifespan of the bees, while addition of brewer's yeast and wort shortens the life of bees. So recommendations is to use supplements supplemental feeding without them, and that the use of these supplements should be more practiced during other seasons, especially if there is no natural pathogen. Thank you in front of my colleagues. Well, I'm quite good familiar with honeybee health and honeybee technology. Well, sometimes honeybee nutrition is a part of honeybee technology, you know. But as you can see, I didn't take a part in that research. So any questions related to that topic, please send to real authors and co-workers. I hope they will be eager to answer on all your questions.
So thank you very much. I would like to ask you to deposit the wish. It might be interesting to know a little bit more about the causes and how they are interpreting these effects. So maybe in the next presentations I might include this. Thank you very much. So we are coming to the last talk before the break. There have several people arrived after my last announcement, so we have a special program regarding the B-book and IBRA, so please do not immediately leave after the last part of the discussion of this talk. So, a contribution from Slovenia, from colleague Karl, and the title is Impact of Sugar Syrup on Lifespans and Age-Related Physiological Condition in Caged Honeybees.
Hello. I'm very happy to be here and to present one part of our research. My name is Maja Smodiš-Kerl and I come from Agricultural Institute of Slovenia. So today I will present you some part of our research with sugar syrups. And first I would like to say some words about background. So the transition from nursing to foraging is known to be associated with food perception, consumption, and so on. And it is known that bees infected with Nosema serrana have higher hunger level, which leads to a lower survival. And in infected bees fed ad libitum and non-infected bees had similar lifespan. But it is not known how substitute energy sources, such as different sugar syrups, affect worker metabolism, including possible influence of Nosema in the midgut, and how they shorten the honeybee lifespan according to season. So there are various carbohydrate supplements which beekeepers use for feeding their colonies. The most popular use is saccharose syrup, which contains white sugar and water. And there are also other syrups in the market which are already prepared to feed the bees. They contain different fructose, glucose, saccharose content, and also different sugar candies, which are very appropriate to immediately use in the colony. So we also conducted field trials. We fed our experimental colonies with syrups. But at the same time, we made lab experiments with caged bees so that we can find out the effect of syrups on the individual bee. So we got our bees from our apiary, and the bees were newly emerged, so we put a comb with a brood in the incubator, and after 24 hours, we got newly emerged bees and we put them into cages. The cages are plastic. These are modified boxes for CDs, where you can store your CDs. But there is enough place for 100, 150 bees. So we put around 100 bees in each cage. And for each experiment, we got 4 to 5 replicates. So we feed the bees with different syrups. We had 4 groups. First group received glucose-fructose syrup, shortly GF, and the second group received saccharose-glucose-fructose syrup, shortly SGF. The third group was fed with fructose-glucose-saccharose syrup, FGS, and the fourth group was fed only saccharose syrup. So to get the seasonal, to influence of season, to maybe we can see if there is any difference, seasonal difference, we made 3 experiments. So in the first experiment we used spring bees, which emerged in April. And in the second experiment there were summer bees emerging in June. And then in the third experiment we used winter bees in October. So they received, first and second experiment, the bees were fed the syrups. And also one cage was just so to compare what happens if the bees are fed with floral honey and bee bread. And in the third experiment, we tried to give the bees diluted syrups. So 50-50 just to see if this step was correct or we made something wrong. So methodology was first we looked over survival, so that we daily counted dead bees in each cage, and then the abdomens were put in the under -20 degrees for further analysis to see how many spores are in the abdomen using hemocytometer and microscope. And then each separate group had also one cage where we collected hemolymph from the bees so that we can see how many proteins are there in the hemolymph, and also if there is any Vitro. This is a storage protein, very important for nurse bees and also in winter bees. And in the hemolymph, we also analyzed the glucose and fructose titer. In the third experiment, we also did some histological research to see if there is any cell death in the midgut of those bees. So, to the results. In spring bees, the survival was over 90% in all tested groups for 10 days. So in 10 days, all the bees, almost all the bees survived. And then there were drastic differences. In the saccharose syrup and glucose-fructose syrup, bees, 50% of bees survived 32 days. And then in the SGF syrup, 50% of bees survived 25 days. And the worst was in the group of fructose-glucose saccharose syrup, where 50% survived only in 16 days. I'm sorry, the picture is missing. I don't know why. Back. Okay, I'm sorry, I don't know why this picture is missing. So, here I wanted to show you how many spores were present in each group. And on this graph, the spore load was relatively low, so it was mostly 1 million, but less than a million spores per bee. And in the fourth group, I want to show you here, in the fourth group, the spore load was very, very high. So surprisingly, bees were more infected with Nosema spores. Just to check whether there is any vitellogenin present in the hemolymph, we looked at day 0 at newly merged bees, and on day 2 and on day 7 in all tested groups, and maybe slightly there was a presence of vitellogenin in newly merged bees and 2-day-old bees, but this is maybe not so significant for this research. So total protein content in the hemolymph in spring bees, we found the highest concentration in the first and second group at 8-day-old bees. There were differences. So just to compare with literature, we can see that in 6-day-old bees, feeding only bee bread and sucrose syrup, they had the highest concentration of protein in their hemolymph, but sugar-feeding bees were the bees that had only 2 micrograms protein. So just to see the whole picture. So the glucose concentration in hemolymph was significant in the second group in different age of bees, and fructose was significantly different between the groups. So the groups had different fructose concentration. So let's see the results in the summer bees. Over 90% of bees survived 10 days in all tested groups, like the previous experiment. And then the saccharose syrup and glucose fructose syrup, those 2 groups had 50% of survived bees in 33 days. So 33 days, bees survived 50%, in 50%. So the next 2 groups, bees survived only 22 days. So there were differences. So Nosema spores, again, in summer bees, there were differences. I'm sorry, here, again, something is missing, the age. Those bees were older bees, so I believe here should be 26 or 27 days old bees when we noticed Nosema spores. And the spores were until million, so it was not a high amount of spores. To see the total protein content in hemolymph of summer bees, we found differences in the third group when they were 11 days old. So there were differences between the groups. And just to check if there is any vitellogenin in the hemolymph in day 1 and day 6, we can see there are other proteins, but vitellogenin is missing. So which is not surprised because bees didn't get protein food. Sugar concentration in summer bees, we found differences on day 16, so between the groups. And in fructose content, there was no significant difference. Just to see the contrast with summer bees that received floral honey and bee bread, we can see that bees that were 19 or 20 days old, they had already some Nosema spores. They lived 40, 50 days longer than the group that we tested with syrups. And as we can see here, vitellogenin is present in 10-day-old bees. So the bees received protein food and they could get this storage protein, very important for survival, for immunity. Okay, and then finally the winter bees. 90% of winter bees survived only 4 days in 4 groups, in all 4 groups. So these bees received diluted syrups. And in glucose fructose syrup, only 50% of bees survived 22 days. But this group performed best, so they reached around 30 days, but only 10%. And in other groups, bees lived a shorter period, 16 and 13 days in 50% of bees. So Nosema spores were all, of course, present, but in very low load. So they were present in all two groups, mostly in older bees. And we checked the ventriculus. We found, we were looking for the peritrophical membrane. It is present in both groups, in 9 days old workers and 15 days old workers. So there was peritrophical membrane. And then we checked apoptosis. So in each cell, the nuclei can die. So cell death can be present normally until 10% to renew the tissue, but if there is too much, more than 10% of cell death, then something. This is indicator that something is wrong. But this results. This is only the beginning of the research. I cannot tell exactly if this is because the bees received the diluted syrup, but maybe this could be the reason because they had too much moisture, too much water in the syrups. So, to conclude, the spring workers. In the first experiment, 50% of spring workers survived 32 days when fed glucose fructose, so the first group, and the fourth group fed saccharose syrup. And we found that the highest number of Nosema spore was found in bees which were fed saccharose syrup. Beta-glucanin was slightly present in the hemolymph of bees, in young bees, newly emerged, and two-day-old bees. And the highest concentration of protein was found in first group and second group of spring workers when they were 8-day-old. Glucose concentration in the hemolymph was significant in 8-day-old bees, and they were in the second group. Fructose was significantly different between the groups. So to conclude, the summer workers, in the second experiment, 50% of summer workers survived 33 days and 22 days in other two groups. Number of Nosema spores was very low, except in the fourth group again, saccharose syrup, where there was over 5 million spores in older workers. So older workers were infected. Significant differences in protein content were found in the third group. Those bees received fructose glucose syrup at day 11. So beta-glucanin was not detected, but it was present in bees that were fed honey and bee bread. Glucose titer was the highest in bees on day 7, and significant between the groups on day 11, so in older bees. And we found no differences in fructose titer in the hemolymph. In winter workers, the winter workers died faster than spring or summer workers. The longest survival was found in the first group, glucose fructose syrup. One of the main reasons could be excessive moisture in the food, so because the syrups were diluted. Nosema spores were insignificantly present in the abdomen of younger and older bees. Maximum number in the first group was found on day 27, when most of the bees were already dead. In the ventricular cell nuclei, we detected multiple apoptosis at day 15 in all 4 groups, and peritrophical membrane was present in the lumen in all groups. So, to conclude my presentation, bees from all 3 seasons survived best, it seems like, on glucose fructose syrup and saccharose syrup, except in winter bees. Nosema mostly infected older bees over 25 days, which were fed saccharose syrup or glucose fructose in winter bees. Beta-glucanin was not detected in caged bees, which were fed sugar syrup, oil syrups, and the highest protein titer was found in glucose fructose and sucralose, glucose, fructose syrup in younger spring bees, and in fructose, glucose syrup in younger summer bees. Glucose content was high in younger spring bees and in all 4 groups in summer bees. But fructose was significantly different between groups in spring bees. Cell bees, infecting these old bees. So our results indicate that sugar syrups could have several impacts on honeybee, not just lifespan, also enzyme presence, maybe also other facts. And for this reason, it is very good to test the syrups also in the field, which we also did, and to compare results, because this project, this examination ended in August in this year, so I was not able to compare everything, but when doing some field trials with syrups and also other supplements, it is very important to see the development, to count how many workers are present in each colony, how is with the enzyme presence and viruses, and heterogeneity level, especially in autumn when winter bees start their tasks, so this is very important for overwintering. So just to introduce our big team, our project leader, Professor Dr. Aleš Gregorc, and from left, our technician Marian Kokal, Vesna Lokar, Dr. Petar Kozmos, young researcher Mateja Soklic, this is our project leader, Aleš. Then Magistra Veronika Kmesel and Mitja Makrst and myself. So I would like to thank to our Beekeeping Association of Slovenia, which supported this research, and also to Ministry of Agriculture and Environment of Slovenia, and which provided fundings, and the European Union. And now I would just like to thank you for your attention.
Line.
So it's just for time for one very short question. Yes?
If you were asked to provide a general advice regarding the best combination to be used in lab tests in cage, what would you say?
So the question was which sugar syrup would be the best to test bees in laboratory conditions.
Yes. Possibly the opinion to provide a general recommendation.
Probably the best would be honey, but honey has many influences. You cannot isolate all the influences. So from the sugar syrup is considered to use it as a control group, but as we can see, Luzima spores develop faster. So I don't know. Maybe I could recommend glucose fructose syrup, because from our results, this is what I could say.
So it was very short, therefore a second one.
Where is live longer? In laboratory or pill treatment?
Survival was tested only in laboratory, so I don't know how they would perform if we put him outside in the colony and back to the... We just took newly emerged bees and put them in the cages, and then we tested how long they were living in the cages, so they were not put outside. In the laboratory, yes.
So thank you very much.